So my name is Nur Ashikin. Next, I will continue presenting on the mechanism of WNT pathway, which is this pathway play role in regulation of various processes that include cell proliferation, differentiation, and also cell survival. So there is two types of WNT signaling pathway, which is this one is with the absence of the WNT ligand. So the inhibitor of WNT signaling pathway, such as DKK and also SFRP, that function as tumor suppressor will contribute to the degradation of beta-catenin through formation of beta-catenin destruction complex that consists of CK1, ABC, axin, and also GSK3 beta. So thus, it prevents the transcription of beta-catenin targeting oncogene. So another one is canonical pathway, which this pathway is contribute to the formation of breast cancer. So the dysregulation of this signaling pathway has been mentioned by Atika, has been implicated in many human diseases ranging from cancer to skeletal disorder. So as for breast cancer, they undergo canonical pathway where the WNT protein are large family of secreted factors that bind to PISL and also LRP5 or 6 co-receptor and initiate signaling cascade. So for the canonical pathway, the canonical pathway is initiated by the binding of WNT protein to the PISL receptor and also co-receptor LRP5 or 6. So the co-receptor aid in the binding of WNT protein to the receptor. So this WNT receptor interaction lead to the recruitment of axin and also this shift protein to the cell membrane and induce inhibition of glycogen synthase kinase GS3 beta protein. So this inhibition of GSK3 beta protein lead to the beta catenin accumulation in cytoplasm and subsequently translocate the beta catenin to the nucleus, where beta catenin then will bind to the LEF or TCF transcription factor and further regulate oncogene such as MYC, CCND, and also MMP7. So as for our group, we will be focusing more on MYC as a target gene to reduce the stemness features of the breast cancer. So MYC transcription factor will then bind to the specific gene sequence and tend to encode protein that supervise the energy production, protein synthesis, and hence promote glycolysis and fatty acid synthesis in tumor cell, providing the tumor cell with substance and energy required for proliferation.